All right, so for last class, we left off on carbohydrates and lipids. We went over these two and the properties that they possess. And we're going to learn a little bit later today how they actually play a role in forming and ensuring that a cell is healthy and able to build itself. And the last two we need to go over. Again, we are going over organic compounds. We need to go over nucleic acids and proteins. And recall that our topic has been metabolisms as of now. And we went over we're going over the organic compounds and we went over some just sprinkled in. We'll learn very specific cases as we progress through the class. But we've gone over what salts are. We've gone over gases, and we learned a little bit about the polarity of H2O. And again, we went over carbohydrates and lipids, and now we got to finish up with nucleic acids and proteins for this packet. And so with that, now another component now, nucleic acids, and how it contributes to the success of a cell is that the nucleic acids is going to be the genetic information, or it's just going to form the genetic information, and that's going to decide what the behavior of a cell is. And the word behavior here is being used very roughly. It's more like the chemistry is behind this. The nucleic acids cause the cell to actually carry out particular types of functions. I had originally mentioned a little bit earlier that every single cell has the same instruction manual for making every single cell in the body, but a heart cell will read pages 1 through 5, and then a brain cell will read pages 7 through 10. A liver cell might read pages 7 too, but will read pages 11 and 12. They all have the same instruction manual, but they read different parts, and they read the same parts sometimes in a different way, so in different combinations. And that's why you can get so many different variety of cells out of the same instruction manual. And so that's what nucleic acids end up serving in the body as the genetic information which will determine the behavior of the cell. And if we examine nucleic acids, you'll see that nucleic acids come in two forms. We'll get into these slight details a little bit more as we progress, but otherwise you'll see two forms. One is famously called the DNA molecule. We're all pretty familiar with this word, but otherwise DNA is going to be the genetic information that is inside of the nucleus. We should make a note of that. In nucleus. Let me get comfier here. I can't write. There we go. In the nucleus. And this particular molecule is unique because it is made out of this sugar phosphate background. Yes, sugar like the carbohydrates that we learned about, but in particular, it forms this like ladder-like system using sugar and phosphates, which you're seeing here highlighted for you. Phosphate group, deoxyribose is a sugar. It sort of resembles fructose, right? That is a pentagon, but otherwise this just keeps repeating itself over and over and over again. And that's what you're seeing here when you look at this strand here, this yellow portion of the strand. And in the middle of it, there is something we refer to as complementary bases. These bases are essentially the language of the molecule, that a particular type of combination or order of these molecules or bases will determine what the actual cell or DNA, what the DNA is going to command to do. And for today, and simply, we really just want to understand how it works together. You'll notice that you see these bases here. You see A, you see T, you see G, you see C, and if you just keep going along, these four bases repeat themselves, and you'll always see the same ones always matching each other. A always goes with T, and G always goes with C. So as you're seeing it in your notes, A always goes with T, and G always goes with C. And so these bases, their names are adenine, guanine, cytosine, thymine, and then this one's an oddball that we're not going to mention just yet, but this is called uracil. And so in the DNA molecule, you find all of these except uracil. You do not find uracil in there. And so these bases are referred to as complementary, meaning that they are always looking for each other. And so we also call the pair or the strands together as complementary strands, because this strand, this half of it, will always match or complement that one. In other words, a T will always be bound to this A C will be bound to this G, A will be bound to this T, and you'll also notice that there's a particular shape to it called a helical shape, a helix shape, as spelled like this, helix shape. And it does resemble as what you know as a spiral staircase, those stairs that sort of circle around as you're going down. There's make you a little nauseous as you're going. They kind of feel flimsy and unsafe, but otherwise they're the ones that circle down to preserve space in for some architecture. And so they are always going to be complementing, meaning that if I were to rip this strand away from the other one, it will be with all the bases will match evenly with the other half. And so that's what we mean by complementary strands. And the only thing there's nothing else to add. Just make sure that you know what bases go with which, and what the backbone is made out of. And the backbone again is phosphate and deoxyribose, which is a sugar, in case you're unsure, a carbohydrate technically. Oops, sugars. And we're going to keep that light today. We'll add a little more detail when we actually learn about when or how cells express genes, or when they're going to divide and why that matters. But we just need some basics today. And then the RNA molecule is going to be sort of kind of like the DNA molecule, but one big difference between them is that it is going to be single-stranded. You're not seeing another half here. But if there was another half, what would be the letter that goes here? What would be the other letter here? What would it be here? A, yes, good. And then so that brings me to the next point. You'll notice that there is no T in RNA. So T, or thymine, does not exist, or you can see the note is already here, only in DNA, but it gets replaced with uracil. And so RNA does not have T, it uses uracil instead. But nothing changes as far as the backbone. It's still made of a sugar phosphate backbone, just like DNA. But the big difference, once more, is that it is single chain. It does not have a complementary strand. And it comes in 3 types, which will be a little bit more clear as we progress or we talk about cell division. And a little bit more about cell biology. But otherwise, messenger RNA is one type, denoted by the little M in front of RNA, messenger RNA. tRNA for transfer RNA, and ribosomal RNA for rRNA. And we'll talk about where we find these, what the difference is, but for now, we just need to know they exist as we progress through the class. And that's what nucleic acids do in the body. So to finish up this packet, we have proteins. Our proteins are going to serve 2 things here, and I'm going to clarify this. We have A and B. And A is going to be enzymes. Sorry, I wrote this last semester and it is very clumsy. Let's fix it today. Enzymes and cytoskeleton. Cytoskeleton. Skeleton, the same way that your body has the bones that make up its skeleton. This little object here, our cell, is not just let me put this in frame, sorry. And so when you look at the cell is not just a little bubble. If I were to put a little needle in here, it wouldn't just burst and everything's just going to come flying out. A needle would just go and seal around. And also inside of it, it's not just like something that you can just squeeze and all of a sudden everything just goes. It actually has fibers inside of it, protein fibers, that actually make up a skeleton that sort of may give it a structure, a frame. And in lab, you're going to learn that they have particular shapes. What are the 3 shapes we find in epithelial cells? What's the flat ones? Squamous. What's the square ones? Cuboidal. And then the last ones? So the difference is that, second lab, you'll get that later today, but they have a skeleton inside that make them and conform them to those shapes. And not just that, they also function like little transportation mechanisms. And I'll look for some videos on that, but otherwise, you'll see some strands. It's a little hard to see, but microtubules is an example, microfilament, and intermediate filaments. These are examples of some of the components of the skeleton, and they are little fibers. And these fibers, they're capable of actually transferring things or transporting them to wherever they need to, like little escalators. And so there'll be things transporting them to wherever they need to go. And it also arranges and organizes all the structures inside of the cell. And they also make very superficial structures that are on the surface of the cell and maintain their shape, like Microvilli. What do microvilli do? Absorb. And yes, so this is again for the lab for later today. So if you feel a little left out, it's okay. It's kind of a preview. And so microvilli are these finger-like projections that help absorb things, but to give it that shape, it relies on skeletal features to sort of give it that shape. And so that's what our cytoskeleton is. Enzymes, very difficult to understand at this current moment, I think where we're at right now, without any more useful photos. But this will be something we'll talk about a little bit further down. And I'd like to sort of think of them as just molecular machines. They carry out functions in the same way that you would imagine Pac-Man is grabbing something and moving it to other places. Or even when we're thinking about this, for now, until I provide some better visuals, is that we were learning about the formation of molecules. And simply A and B combine to make a molecule AB. And so enzymes would grab this, and then all of a sudden together, it'll carry out the chemical reaction required to synthesize, to do that dehydration synthesis, and then combine them. So they are like machines that look for their ingredients and then bring them together, and then they carry out the reaction required to actually unite them. And not to that same effect, you also have enzymes that go look for these molecules. Let's say that you're consuming some nutrients. Your nutrients, we saw that your body needs to break them down into smaller monomers, to smaller pieces. And so sometimes it'll grab the whole thing, and then that enzyme will carry out the hydrolysis reaction that we need to break things apart, and it'll actually break it apart. And so they're like little molecular machines that actually carry out these reactions. And sometimes their role is just to transport things from one location to another location. As simple as that sometimes. And when we get better photos in a future packet, because I might have to add a page to one of your packets, we'll talk about it then. But otherwise, structure now, or what they're made out of. So as we look at proteins, they are made of components called amino acids. Amino acids. These are considered the building blocks, and there are 23 total amino acids. All proteins are technically all going to be some combination of these 23 amino acids, and depending on which one it gets determines what it does. And that's a complexity we'll get into maybe a little bit further. In the summer, I might reduce the material just to make it a little bit more manageable. But otherwise, as we look at an amino acid, you'll see that it has some regular atoms that we've already sort of looked at on the periodic table and saw their valence shells like oxygen, carbon, hydrogen. They're all these common ones. And you can see a covalent bond, a double covalent bond. And all amino acids have a carboxyl group. They all have an amino group. Whenever you see the word amino, it typically implies nitrogen. Physiology will traumatize you on a lot of this stuff. I will not have to. And so the carboxyl group is again going to be some combination of carbon and oxygen. So that's why you're seeing carbon and you're seeing oxygen on both ends there. Carboxyl group. And then it always has this little fun hydrogen just hanging around there. And all of them have it. Absolutely every 23 amino acids that you see have this same exact component. A carboxyl group and this hydrogen there, and an amino group. And this central carbon. What's always going to have some variation is going to be what we're seeing here, this R group. This R group is considered a side chain, and what makes every amino acid different, even though they all have that same component, this part here, this R chain will be different among them. If you're a certain type of amino acid, you might have more carbons this way. You might look like this. Or some of them might look like this. Like they will have a full-blown ring there and some other structures. There may be very short branches that just branch out very differently. And it's all just going to be carbon, 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 carbon. And sometimes they just have much more complexity that we don't enter in this class. But that's what really makes them unique, is this R chain. It's what gives every single one the ability to manipulate the shape of something in its own way. And so the relevance for today is that when we form a bond between them, and I want you to just quickly look here, if you're seeing And let me make sense of my own picture here. Oh, yes. If we're seeing we want to combine this amino acid, alanine, as an example, you can see has a very small R chain. All it has is a CH3. That's it, just a carbon, 3 hydrogens. Here you can see that glycine has just another hydrogen, nothing special about it. But you can see that these two can combine together. And what type of synthesis combines things together? And you can even already showing you a clue here. Dehydration, because you can see now that alanine and glycine will combine together. And you can see here there's no connection, but here we have our bond. And what happened? H and OH was removed to form a water molecule. Therefore, if we lost our water molecule, we remove the water molecule and we synthesize, we build up, then it's a dehydration synthesis. When your brain, you shouldn't learn dehydration, the word dehydration by itself. You should learn the word dehydration synthesis together, DS, to always remind you which one forms, which one breaks. And the one that forms is the one that dehydrates. And then the one that breaks apart is called what? Hydrolysis. And so we refer to these bonds in proteins as peptide bonds, as written above. Peptide bonds. They are covalently bond together. They are not ionic. They are sharing electrons. They are not giving up or accepting electrons. They are sharing electrons. And simply for our slides here, dipeptide means that you have a peptide that has how many amino acids? Nice and easy. And many more than 2? Poly. Just like when we saw polymer or polysaccharides, disaccharides, you can see that the same type of reactions are required. Dehydration synthesis or hydrolysis to break it apart. But otherwise, nothing really changes overall. And here's a tripeptide, which can also be named a polypeptide because it's more than 2. And again, to form, this is a connecting. They lost water to make it. But otherwise, that's really it for this slide. Just basics on proteins and amino acids. Now, the complexity of these proteins, once more, if you look at an amino acid chain, which is nothing but amino acids chained together through their peptide bonds. These peptide bonds, as you can see, can be very, very long. And again, here you can see the R chains are going to be different. We don't know exactly what amino acids they're using here. But again, they can be as short, like we saw the previous one, just a hydrogen. They can be a carbon with hydrogens attached to it here. Or they can be incredibly long chains. We don't know what they are. We just do not know. But whatever combination you have will eventually cause very unique folding. And so, before we get to that folding, a single understanding of the chain of amino acids, the amino acid chain, is called a primary structure. And so a primary structure is defined as the sequence of amino acids as written there for you. The next structures that form as a result of whatever sequence you got, because again, the R chain will determine how it folds, is that we actually see that it yields very common shapes. Those 2 of those shapes are going to be called alpha helix and the other one beta sheet. And an alpha helix does exactly what the helix structure looked in a DNA molecule, in that it forms sort of a spiral. Oops, wrong thing. And so that's why it's called an alpha helix. And so again, you're just seeing that A1, A2, A3, etc. You're seeing A1, A2, A3, A4, A5, A6. You're just seeing them fold because these R chains are interacting with each other in a very unique way, causing this sort of tension, this sort of binding that causes this shape. So once more, alpha helix or a beta sheet. The sequence may cause it to form some sort of sheet-like structure like this. And we consider the alpha and beta sheet secondary structures. Secondary structures. If we look even larger now, when these two can combine. Alpha helix can combine with a beta sheet, There might be a little bit of a chain in between them, but sometimes they can directly attach, alpha helix and beta sheet, and you can see them here now. You can see an alpha helix, and we're not really seeing our beta sheet in this case, but you're seeing a helical structure. A helical structure we're highlighted here in this box. And then we're seeing a very small one here, and we're not seeing a beta sheet in this particular example, but otherwise there may be a beta sheet included sometimes added in the space, and there might just be some small amino acid chain linking them together. And then we consider them uniting alpha helixes with other alpha helixes, or an alpha helix with a beta sheet, tertiary structure, which is a third level or third degree or a third level of organization. And then we start seeing a more three-dimensional structure when they all combine together. And then finally, we have our quaternary structure, which again implies 4. And because we see that a lot of these are the more popular ones that we're going to encounter in this class, this is an example of a protein structure. You're seeing that it has 4 units. Here's one in blue. Here's this blue one. Here's a purplish one on mine. I know you don't have color, but purple on mine. And here's another blue segment. And another purple. So you can see 1, 2, 3, 4. You can even see at its very center, it could possess particular sites that are responsible for holding or finding molecules. This famous example here is hemoglobin, and it'll matter for us because hemoglobin carries oxygen. And so blood cells are loaded, the inside of their cells, they possess a lot of these proteins, these hemoglobin proteins that carry around oxygen. And they use these little sites called heme units to actually attach the oxygen, and then now they travel throughout the red blood cells and they deliver it throughout the body. And keep in mind that I mentioned that we have proteins play 2 roles. The formation of enzymes. Hemoglobin is considered an enzyme for the most part, a carrier enzyme, a carrier protein. And so we're going to, for today, just consider it the same, a little machine that carries things around. And we also said it forms the cytoskeleton component of things. And so collagen is an example of a structural part. So the collagen would be an example of something that makes up the cytoskeleton. And again, hemoglobin will be a little machine. We're going to just keep it simple, a molecular machine. And it carries around oxygen. So we're seeing 2 examples of how these proteins can come together to form either a structural component or a protein that actually carries out just a machine, a molecular machine. And that really summarizes our 4 organic compounds. So this is something you have to log away, that when I say, what are the 4 organic compounds, you should be able to list them and know the like properties that we talked about, or the introductory properties we learned about. Luckily, since this is anatomy, we don't get too crazy into the molecular basis of these things, but when you get to physio, this is enough background to keep you safe and enter very strong. And so I think that we are absolutely done with this packet. Yes, we are. And so to summarize, keep in mind that this packet was about how cells themselves, these organisms, all require metabolites. They're going to do chemical reactions, whether it's for the formation or breakdown. They're going to use it to make energy. And remember that only when you break down molecules, we form energy. We form energy. And they're going to use it to carry out very important functions. All right, next packet. That's going to be the one that looks like this. One second, let me find it. Here. Okay. I do not know why this happened. Let me just quickly fix this for myself. One second. If you're watching this video now, you can skip ahead. a few seconds. And okay, there's one more extra added in there for some reason. Okay. All right. Okay, well, I'll fix it later. Okay. And so, let's move forward. So now we're going to go into our introduction to cells. And so we have to become acquainted with something referred to as cell theory. And so cell theories, if we just look at our illustrations here, is that cells are the building blocks of all living creatures, of plants and animals. And so we can see that your tongue is going to be made of a particular type of flat cells that are stacked. For lab number 1 today, what is these stacked cells that make the inner linings called? Anybody remember? First, what's the shape of the cell? And they are built on top of each other, so what word matters? So what is this then together? Stratified squamous, good. And that's for the recording to help you all refresh your memories on that, or make sure you can see the overlap. And so they build up everything. Everything in your cell is made out of tiny little individual cells. As you look at a lot of these schematics, they're always roughly drawn with a boundary, which is going to be the cell membrane, the wall on the outside of the cell, and on the inside it'll have a nucleus or a center that commands or tells the cell or controls the behavior of the cell, like so. Cell membrane, and again, the nucleus on the inside. And they are capable of dividing. And so we mentioned this the first day, that cells, they may make themselves larger. They make copies of everything they have on the inside. They get larger because of that. And then finally, they divvy up everything they have inside. They divvy up all the internal components into 2 equal ones, and that's how they create more cells. So even in this case, these cells may divide and create brand new ones that just clone and go and migrate to the very top to create more cells. And we'll discuss that a little bit more in detail later. And then, as we all know, they carry metabolic functions. And so this is just a rough drawing of a cell. They take in nutrients from our digestive tract. They take in gases from our respiratory tract. And together, they carry out their own metabolism to carry out reactions that either break or form things. And then eventually, they all produce a little waste. They all produce a waste gas. So after the utilization of oxygen, they end up producing CO2. And you can think about it in that manner. And then also the waste, after using those nutrients, they have to produce a poop, a waste. And so they also, in essence, can eat, and they also produce a waste, just like the large organism that they're attached to does. And so this should look slightly familiar for some of you today, and it will look familiar for half of you later, that when we look at our egg cell, this is our egg cell, and this little tadpole one that's in here is a sperm cell. This is from the male, biological males, and this is from biological females. When they combine together, their genetic information is going to mix together to make a full human being eventually. And so at the start, when these two find each other, the egg and sperm cell, they eventually cause a bunch of cell division where those two just make all the combined DNA, to keep things simple, the combined genetic information to make brand new little cells that will eventually turn into the entire human being. The entire human being is made out of these components. Epithelial tissue, it's made out of connective tissue, made out of muscle tissue, and made out of neural tissue. And so we saw in lab today, we learn about epithelial tissue, we'll learn about muscle tissue, and we learn about nervous tissue, and we leave connective tissue for next week, or next lab, next class, both. And so these are the components that make up an entire human being, but they start off as sperm and egg, and they just undergo cell division. Eventually they branch off, and when they start branching and becoming different, they're undergoing cellular differentiation. This is a word we have to understand. It means to actually transform or turn or specialize, to go from basic little cells to eventually become very specialty cells. Where are we going to encounter neural tissue? In the brain, in the spinal cord. What's an example of a muscle tissue cell? Right? The skeletal muscles that we learned about in the organ system, skeletal muscle, the cardiac muscle, smooth muscle, and then epithelial, a little bit tougher, but very simply, the ones that you encounter on the surface of things. Like your skin is an example of epithelial tissue. Epi, what is Latin for epi? Epi Latin for? Above. So you can see that epithelial tissue means tissues that serve as surfaces. And so it's kind of a little preview of what you'll experience a little bit later. But otherwise, they specialize. So when a cell becomes or goes from being pretty generic to becoming specialized, we refer to that as cellular differentiation. One last word to introduce here, and I do want you to know what it is zygote. Zygote is just a Latin way of saying that when a sperm and an egg meet together and fuse together, they are in the position to actually begin cell division. So zygote is like saying a fertilized egg in a way. A fertilized egg, meaning that a sperm and an egg have formed a union. Ask away.
What's the difference between a zygote and a gamete?
A gamete is to refer to as the sex cells. We've only used the word sex cells, and in males are the sex cells is sperm. And in the females, what's the female symbol? Is it a little plus? Yes. And then what's the male one? Like an arrow? Yes. And then the sperm form, and then the egg is for female. So sperm, males produce sperm, females produce eggs, and that's what the word gametes is. Gametes is the appropriate term to imply the sex cells, gametes. So to use the language, when two gametes, when the male gamete and the female gamete come together, they form a zygote. That is the appropriate way to use those terms. All right. And a new term to sort of touch on here. All these cells coordinate in the body, their metabolism, their activities, they all coordinate and regulate and balance the body's processes. When the body is talking about balance, and we're going to break this term a little more in detail, it's called homeostasis, and this is maintaining the appropriate amount of materials being made by these cells, the appropriate temperature in the body, the appropriate pH. A lot of things are encompassed when we talk about the word homeostasis. So for us, we're just thinking about coordinated activities allow a balancing of the body's processes and functions. Or they all coordinate the activities for homeostasis. And here, this is a little bit more, just more of a verbal description of what I mentioned, but otherwise, a fertilized ovum, this is another way of saying that an egg that's been fertilized ovum. And now these cells can become anything. And now a little bit of a fun fact, a little bit of a break in a fun fact is the concept of stem cell technology is based off of this premise. That, recall that I said that a human cells, and we're going to ignore this for a second, but when we're at this level, these cells are capable of turning into that whole families of tissue types that we saw, epithelial, connective, muscle, and neural. These have the potential to turn into absolutely everything. That's what stem cell technology is, stem cell treatments and biology is that these cells can turn into everything. So what stem cell biology was is that usually donated sperms and donated eggs were fused together in a dish, and then when they go into this stage, you can grab these cells and you can place it in the environment that these cells are used to, and they will turn into nervous. You can turn them into muscle. You can turn them into every type of cell because they have the potential to turn into absolutely every single thing. And during the George Bush era, because we do live in a very politically and religious world, that it was removed or banned, or all science was banned that would destroy sperm and egg, even if it was out in dishes, or in vitro we called it, or in dishes, even though it wasn't in the body, because it was assumed that life begins when sperm and egg combine together to form a zygote. And so it was completely banned. But scientists, knowing that all the genetic information, it doesn't matter if it's a nervous cell, muscle cell, connective tissue, and an epithelial cell, we know that the instruction manual is the same for every single one. This nucleus instructions manual, the DNA molecules, are the same as the DNA molecules in here. But again, they're reading different parts of that instruction manual. And so what we do is that we create genetically modified viruses, and then we force it to read the instructions that this family has to read. We force it to read what these have to read, and then these cells, more than likely, we would turn these cells into nervous. We would mostly grab some of these cells. We would grab these and turn it into this. We can grab this cell, turn it into this, and we don't actually have to destroy sperm and egg, but it's just a sort of a, crazy to think about, right? And even in so much that, you may have heard like some news online that two females can actually have or produce a viable offspring, because you can actually grab one of their cells and you can actually convert it, or grab half of I don't want to get into too much complication, but you can actually turn it into a viable zygote. And there's a complication to this. You know what, I'm getting a little out of a tangent here, but otherwise that's the premise behind stem cell biology, is that the nucleus contains the information to form every cell, so we go backwards now. And then we create these. So we grab these, transform it to this, or we can also make it turn into this, and then we can make it turn into this. And so that's sort of a unique or fun little fact about stem cell biology or stem cell technology. If any of you never know where you're going to end up, so it's pretty good to know.
Professor?
Ask Glenn.
I have a question.
Mm-hmm.
Is that the same thing as when you take out cells and then you put them back into your body to reconstruct?
Yes.
Like if your calcium is starting to disintegrate, can you do that?
Not necessarily calcium, but I will say, like, for example, blood cells, like white blood cells, like a project that I was involved in a few years back was that I was grabbing white blood cells, right? Like white blood cells, they're floating in your bloodstream, and if anything dangerous enters your bloodstream, they go and they just grow and destroy and attack in one way. And so there's a problem with that, though, that if I were to grab my white blood cells, and even though they do the same thing in your body would say, hey, these are my white blood cells, and then they'll start attacking your own body for that reason. So we were trying to develop stem cell technologies where we would actually grab, say, white blood cells, and then we were trying to turn them backwards into this, and maybe turn it into this. Okay, let me make it an even easier example. If I grab, let's say I want to study your brain. If I open your skull and I take out those cells, they might grow. Those are very hard cells to culture. But if you did, it's incredibly detrimental to your own health to do that operation just so that I can maybe get those cells. And so what we can do is that we can grab something easier, like something from the lungs, and then we can turn it into this phase, and then I can force it to turn into nervous tissue. And then inside of that dish, they will look exactly the same way that they look inside your head, because they come from your body. So if you have a genetic disorder that makes your nerve cells wonky and funky, if I turn your normal cells into your brain cells, because it's a genetic problem in the brain, they will look wonky and funky in the dish. And so then I can study the disease inside of that dish, or I can throw drugs at it and see if it alleviates the disorder in the dish first. And then so that's the power of this kind of technology, is that we can work with body parts without working with the entire body. And then eventually we move on to animal studies to actually see if any of those things work. And so that's an example. And calcium is a inorganic, or like a salt. So technically, if you might having bone cells that maybe are not working at producing calcium, one day there might be a scientist that will produce bone cells that can actually make you adequately produce calcium. That's something that I can see as a possibility.
Periodic tables in calcium and organic alloy.
It is, and it is. That's why it is not a cell. It is a component or a thing that the cells use. And so, yes. And we'll actually get into calcium in a little bit as well. All right, we got to keep moving because we got an hour and 12 minutes. All right, so now once more we're seeing another little fun schematic, and my advice for all of you is to start here when you're studying your study for your cells, because what you're learning right now, you have to separate from a person, right? We're learning anatomy, we're learning about a person, but right now you want to just think about the animal that is a cell. And a cell has tiny little components on the inside. It has little organs, and so those little organs are called organelles. Remember in Latin, typically when you're adding some sort of prefix? This is not a prefix. Suffix? When you're adding a suffix, when you add something at the end of the word, sometimes it implies a variation of that word. So like organ is like you all know an organ as a heart, stomach, small intestine, but organelles means little organ. And so right now you're learning about the animal that is the cell. So separate from human anatomy and we're thinking about cell anatomy. What is this little creature made out of? So some terms we're going to start from the outside in. The fluid outside of a cell and what marks the boundary is going to be the cell membrane. In the lab, you hear me say cell membrane a lot, but now I can use plasma membrane because that'll be more appropriate. And plasma membrane is going to be the outer membrane or wall of the cell. The word membrane means some sort of boundary. And so this is going to be the boundary of the cell. There is an outside and an inside. And so as you look at this word, extra means outside. Cellular means exactly what you think, cell. So outside cell fluid. If you look at this one, intra means inside, inside cell fluid. So there's fluid outside, there's fluid inside, and the only thing keeping them separate from each other is this plasma membrane. The word cytoplasm should in your heads mean area around nucleus, and we'll leave it at that, around nucleus. And let's talk about cytoplasm for a second in this picture. And you don't have to flip pages, I got it for you. And here, when you look at this segment here, this is your nucleus for now. We're going to address all these parts, but first, this is your nucleus. In the nucleus, just like the atom, it meant to imply the center, soul, or heart, whatever determines the properties of whatever we're dealing with, the nucleus. And so the cell itself has a nucleus, and this nucleus houses DNA. DNA is stored, or the genetic information is stored inside of this nucleus. So there is boundaries to this. Outside of the nucleus, everything outside, and let me draw it in here, everything outside of the nucleus is called the cytoplasm. It is the area around the nucleus, and it ends at the plasma membrane, and it does not include the inside of the nucleus. It does not include inside. It's called cytoplasm, meaning outside of the nucleus. Does not include the nucleus. Cytoplasm. And so you can see that our flow chart now makes a little more sense because cytoplasm is going to include everything outside of that nucleus. What is outside? Cytosol. First, let me introduce you to a new Latin word that's going to haunt you for a while. Cyto. In your heads, and actually is cell. It means cell. Cyto. Cyto. You may see some variations of the way it's used. For example, you may see the word adipocytes. And I had you all learn a word. What does chondri mean? Cartilage. So what do we think our cartilage cells are going to be called? Chondrocytes. 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 So you can see cytes means cell. Chondro means cartilage. The word itself means cartilage cells. So you'll see this theme come up over and over again. So you must be able to recognize when you're being told this is the name of a cell. And so this is a light example of that. And so sol in Latin typically implies liquid. And so this word means liquid. And I'm just going to put cellular liquid, right? That's what the word technically translates to. cellular liquid. Cellular liquid. And organelles are going to be all those little tiny organelles, or the little organs that we saw. There, you can just even refer to it as the tiny organs. And this is our goal today, is to break these down. This packet can be summarized essentially with this image. These are the organelles that you must become familiar with over the weekend. You must learn what these organelles are and what they do. And your homework has a lot of problems to help you study for that. And so let's get into it. Anything I'm missing? Let me just double check. There is this word interstitial fluid, but for now, you can cross it off from your notes because it's going to come back on the next packet, and it's much more easier to understand it a little bit later. And so nothing changes though. Extracellular fluid, intracellular fluid. And so there are interchangeable words, right? The fluid inside the cell was called intracellular fluid, or again, cytosol. And so the plasma membrane, by its definition, or the fact that it's a membrane, something that separates something, separates contents, is that it separates the cytoplasm from the extracellular fluid. All right, so just reading through this and making sure you understand the terms is the easy part for this. Now, your organelles come in two forms. Some will have a membrane and some will not, hence non-membranous and membranous. All right, make sure that you know which ones are considered membranous and which ones are not. As you get into each one, it'll be very easy. You might think, oh, I got to actually learn the categories. You'll see as you go through each one, you'll be like, there's no way these have membranes. It'll just make logical sense as you progress. And so let's get into it. Let's get into our tiny little organelles. And hopefully I have time to show you a little video at the very end. I just want to double check our notes. Perfect. We'll have plenty of time. All right.
Have you been your salsa dancing?
No, not yet. Let's wait till I finish the competitive routine. I'm still learning how to do a damn backflip. Dang, it's on the recording. I will show you when it's done, and hopefully I have it done in a month. So we'll see. Because I'm the next champion. Watch. Anyways, so if we keep going if we look at this cell here, right, this cell has components. We're going to start at our nucleus. The nucleus here is going to be this portion of the cell that houses the DNA. And your notes, I think when I made them, I accidentally put it in the wrong segment. I wanted them to go in order. So I'm gonna have to move around a little bit. But nucleus, its structure overall is that it has its own membrane. Remember that it's one of those in the category that has a membrane. We call that membrane a nuclear envelope. A nuclear envelope. And it does something that you already are familiar with. It controls metabolism. These cells, when we say behavior in a cell, they don't behave, like humans. Like, we're not talking about the same type of behavior. We're talking about what do they do at a chemical level. And so if we're talking about at a chemical level, we are talking about reactions. We are talking about metabolism. So they control the metabolism of cells. The metabolism of cells. And also the genetic information, your genes. They contain a little bit of mom, a little bit of dad, all the time. They all possess a little bit of mom, a little bit of dad, right? And so that's what I mean by genetic information. And we'll elaborate on that a little bit further when we talk about cell division. And so at an offset, what we need to know just right now is that it also has its own fluid inside. And we saw the fluid inside the cell was called nucleoplasm. And so, cytoplasm inside the cell. That's the fluid outside in this space, cytoplasm. But if it's the fluid inside of the nucleus, it's simply called nucleoplasm. So you can see that plasm tends to refer to fluid. And this comes back to haunt us. Log it away for today. Plasma implies a fluid or liquid component of anything. Plasma. And enzymes are What family of organic molecules? Proteins. And you can see enzymes, molecular machines, and also proteins as structural components, right? And then DNA and nucleotides. Now, this nucleo implies the nucleus. And what are these nucleotides? I'm just going to quickly go back. You don't have to find your other packet. Just want to make sure that I go full circle here and we address everything. That when we looked at our bases, these are nucleotides. The bases that we learned about, these are referred to as nucleotides. It includes those nucleotides. All right, so you might want to just write that word down. That, or bases. On the other packet, just write bases to remind you. Bases. Bases. For the nucleotides. Bases. Next. I'm going to go in order to what will help us with some visuals later. Next, we have our endoplasmic reticulum. Endoplasmic reticulum. A lot of times you'll see it shortened as ER. And endoplasmic reticulum comes in two different forms. But just looking at it together, what it tends to be is a network of sheets and channels. So if we look at our illustration, you can see endoplasmic reticulum comes in two forms. One is called smooth, one is called rough. Very easy to learn. This word alone is hard to learn, but learning which one's smooth and rough is easy, and their functions are easy as well. Because if you just look at the illustration, you can see that the rough one has a bunch of little studs or a bunch of little freckles, right? A bunch of little dots, bumps. We'll learn what they are in a bit. And that's ribosomes. And so they start off as rough. That's why we call that rough. And so you'll see that there are other ones without them at all. And so those are called smooth ER, smooth. So those are the two forms, and they do two different functions, but we'll talk about it right now. Altogether, something that they do is that they are creating the products that they're going to secrete. And, it's not that fancy. We're going to use a very non-fancy example for now. But what's one thing that they love to secrete in, like, the intestines, for example, lab number 1? Mucus. So we're just going to pretend that when we say secretory products, mucus is an example. Mucus. That's right then. Mucus is an example. It's more relevant, and we'll see it come up over and over again. Mucus. And it can either it can store things and transport things, and it can also detoxify drugs. When you consume drugs or alcohol in your liver has extensive smooth ER to help you detoxify anything that you need. That's why you'll notice that there are diseases that occur with alcoholism. Does anybody know what they're called by any chance? Starts with an S? Cirrhosis. And so that's because the liver works overtime to try to remove all that toxin or the alcohol, the byproducts and the alcohol. Just keep it simple, what alcohol does to the body. And so smooth ER is responsible for the detoxification. And let's go to this next page because I do remember I split into this segment here. And so you'll see smooth ER again once more defined because it doesn't have those little dots. Those little dots are called ribosomes. We'll talk about what they are next. Ribosomes. So smooth ER does not have those little tiny beads, or again, the ribosomes. And it synthesizes lipids and carbohydrates. And we know what lipids are, right? We know what carbohydrates are. And again, they're synthesized in the smooth ER. Rough ER is a little bit different. It has ribosomes. And ribosomes, the role of ribosomes, as you're going to see in the next following segment, is that they synthesize proteins. They synthesize proteins. That's what ribosomes do. They synthesize proteins. So we know the function of rough ER. We know if they have ribosomes, then what does the whole rough ER do? Synthesize proteins. That's all. Because if it's rough, your head should jump to the logic, it's rough because it has ribosomes. If it has ribosomes, we synthesize proteins. And then we have how many molecular compounds? We have 4. Nucleic acids, DNA. Where do we find DNA? Nucleus. So we don't care about it anymore. We already know where it is. So now we're left with 3: carbohydrates, proteins, lipids. A smoothie of the ER, which ER deals with proteins? Rough, so that gets rid of this one. So now we're left with 2. And so what synthesizes lipids and carbohydrates? The smooth ER. So the smooth ER deals with the last 2, right? So you can see that all 4 have been addressed. We've dealt with proteins. It's the ribosomes that are attached to the rough ER synthesize proteins. Lipids and carbohydrates are synthesized by the smooth ER. And then DNA and RNA, not really involved right now. Not right now, no. We'll be in a second, but not right now. And so we've touched on DNA. And what was the other type, the single-stranded one called? RNA. So you may see a note here that says rRNA and mRNA. And so, jumping into that note, ribosomes, their function is to produce proteins, synthesize protein. But what is relevant now is that when we look at our rRNA, right, that was one of the types of RNAs we learned about. We learned about mRNA. They are associated with ribosomes. That's all I want you to know for today. Associated with ribosomes. And we'll break it down when we come back next week on Monday. We'll talk about those things once you've studied it over the weekend and you're very familiar with these functions and you're very fluent with them. Then we'll get into a little more details. But I'm just giving a home to absolutely everything we've learned so far. And so with that, if we know ribosomes are attached, they are going to produce proteins in the rough ER. Now, this comes with a little bit of a new fact that we have to be careful with, is that if they are fixed, this is the same word as attached. If they are fixed, that means that they are attached, and what attaches ribosomes? You already know, the rough ER, the rough endoplasmic reticulum. And let's actually, for consistency's sake, add it, rough ER, to the rough endoplasmic reticulum. And they also come in a free floating form, meaning that they are not attached to anything. They're floating around. Hence, if you look at this image, you can see obviously these are attached. But then you'll see some ribosomes that are not attached to anything, and they are floating within the cytosol, within the cytoplasm. And now I want you to also, now that we're here, I want you to become aware of what the difference between cytoplasm and cytosol is. The cytosol refers to just the fluid within that space. The cytoplasm refers to the area and everything in it. So you can see that the cytoplasm has fluid and the organelles. They are both part of the cytoplasm. This is sort of already indicated by your umbrella, but I just really want you to highlight that this is the fluid part, this is the little organs, but all together, the cytoplasm is talking about both of those components. And so you can see that sometimes I play with the language, or I just say it in different forms so that you can hear it, but scattered in the cytoplasm. That means it's scattered in the area all around that space, but I can also say scattered in the cytosol, and you should still understand what I mean by that. All right. And these notes, these are things that we come back to build off of. So let's neglect it for now to keep things simple and just keep moving fluidly. So now we see our nucleus, we've seen our rough ER, we've seen our smooth ER, we've seen these little ribosomes. Now we got to learn our Golgi apparatus. Golgi apparatus. Our Golgi apparatus is going to be this one here, Golgi apparatus. Sometimes you'll see it called Golgi body. Both are acceptable terms for it. And this is going to be another membranous stacked sheet. And let's go to that segment, Golgi apparatus. And they're going to be stacked, flattened membranes. And what they do is that they store, alter, and package synthesized, and it got cut off on me here, but proteins and lipids. Let's add that here. Note. It got cut off when I added it, so let's fix it. Proteins. And lipids. And again, we just went back one page in case you're not sure. Here. Store, alter, package, synthesize proteins and lipids. So if you think about it from a simple perspective, proteins were made by the rough ER, and then they're going to be packaged or stored or modified or completely transformed or fixed in the Golgi apparatus. So there's an order to this stuff. The DNA or the nucleus tells the cell what to make and what proteins to make. The rough ER will make the proteins, and the smooth ER will make lipids, and then they're all going to go to the Golgi apparatus, where they're going to be sort of altered and processed, and they're going to be checked, and they're going to be refined and made functional, made well. And then the Golgi apparatus, the role of the Golgi apparatus is to deliver them to the surface of the cell, to the surface of the cell. And what's the product that we're going to focus on for, like, what's a product that's made by cells? No. More simpler. What do cells secrete? Mucus. And so it's an example. Let's pretend that the proteins were told to make mucus components. And so you can see that they're going to go into our Golgi apparatus, and then they're going to transfer to the surface of the cell, and then it's going to deliver that mucus out to the outside environment. And so, after that, sorry, you don't have to flip. I'm still double checking. The rest of it is easy. Okay, nice. And so that's our narrative. Nucleus, rough ER, smooth ER, to Golgi apparatus. Golgi apparatus will send things to the surface of the cell. Its role is to transport things to the surface of the cell. Now, narrative number 2. There are 2 types of ribosomes. There are some that are fixed, and there are some that are free-floating or free. If they are fixed, that means that their destiny is to go to the Golgi body or the Golgi apparatus. Therefore, the job of the Golgi body is to deliver it outside of the cell. So what do free ribosomes do then? Because only the rough ER is really responsible eventually for sending things to the Golgi. So the free ribosomes, they produce substances and materials, proteins. They produce proteins that are going to stay inside of the cell. And that's the fundamental difference between fixed and free ribosomes, is that free ribosomes produce proteins that are going to stay within the cell itself, within the cytosol. And their job is to serve as little machines. Those proteins are going to carry around stuff, move stuff, carry out chemical reactions that maybe even some of those are going to go inside and control DNA, but their job is to produce proteins that are going to stay inside of the cell. So maybe we should add that note. Here, where we see free produces proteins that stay inside cell. And fixed will eventually be secreted or go to the plasma membrane. And now you're seeing why I have this note here. So what you can do is do this little bracket like this. And that's why that's there, is that the fixed ones will eventually send it to the Golgi body. Eventually, that's the job of the endoplasmic reticulum, is that once it makes the proteins, it wants to send it to the Golgi body. Therefore, if it's going to the Golgi body, it's targeted or it's meant for secretion, or it's going to go to the plasma membrane. Or it's going to go to the plasma membrane. And we will come back to this fact one more time today. But otherwise, the free ones, they produce proteins that stay inside the cell to carry out the functions inside the cell. And now, you have little vesicles, lysosomes and peroxisomes, and they're going to look like these little vesicles that you're seeing here, like little bubbles. And there's something that you already are familiar with. They are your, when we talked about lipids, lipids themselves form these tiny little vesicles. They love to look for each other, especially if they're like each other. They look for each other and form these balls, these little bubbles in essence, and they can store stuff inside. And this stuff that's stored inside will be separate from the contents outside. And that's because, again, they have these hydrophobic tails, and they have this hydrophilic or water-loving segment outside. And so the fluid is separate from the contents on the inside. And there's a good reason for this, because if we look at the structure that we're on now, these lysosomes and peroxisomes are essentially little lipid missiles. They're little lipid containers. And they possess things inside that could be harmful to you. And an example, or the start, lysosome are these vesicles that are made of lipids that contain digestive enzymes. They can break things down. Again, enzymes are little molecular machines that are made of proteins. Dang it. Made of proteins. Little molecular machines that are made of proteins. And their role is to, again, digest things. Therefore, you can see that their role is to break down a lot of compounds, organic compounds. What are four examples of organic compounds? Proteins, carbohydrates, lipids, and which one did I say? Nucleic acids. And they also can destroy organelles and invasive pathogens. Invasive pathogens. So that stuff that they possess inside, the digestive enzymes, are dangerous to you because they break and destroy these when they're old, damaged, it destroys it. So they need to be separated from the components outside. And that's why these lysosomes are these little vesicles that contain or separate contents from the rest of the cell. Let's go ahead.
One little thing that I personally use to help me memorize that, I think about because it's like a cleanup cruise. I think Lysol.
Oh, that's good. Yes, lice in general means in Latin to break things apart. We've already even experienced a play off of this word. Where have we seen lysis indicating a breakdown? Think of the word. Sort of a nice little review. Hydrolysis. So lysis, Lysol, though Lysol is a chemical that can break a lot of contents like smudges and contents through just chemical reactions that are very reactive and are capable of removing organic compounds and stains like oil stains, scum, like little like fungal scum, things like that. It removes organic compounds because it's capable of lysing it. So yes, Lysol is a fluid that breaks things apart. That's why I said sol, like cytosol, is the fluid of a cell. Lysol is a fluid that breaks things apart. That's where the name comes from. And so lysosome will definitely help you, or Lysol is a good way of remembering what lysosomes actually do. And to that same effect, we have peroxisomes. This has something that you're all familiar with. You all had a scratch, and what do you add to it when you have like a cut? What do you do when you get a rash or a cut when you're young, when you fell and you get a scrape? What do your parents add to it? Hydrogen peroxide. And so these somes, or some is always going to imply some sort of lipid vesicle. You'll see this throughout different science courses. And lipid vesicle, so some, vesicles, lipid vesicle. And so peroxisome is a vesicle made of lipids that contains hydrogen peroxide. And it's great at breaking down organic compounds. Organic compounds. And so you can all imagine that when you add hydrogen peroxide, we all have that cut and you add it and it stings a little bit and you even see it bubble and sizzle, right? And it hurts. That's because it's killing absolutely everything there. Hydrogen peroxide is toxic to everything it touches. It kills everything that's living. And so when you place it on there, that's why it hurts, because your nerve cells are like, what are you doing? It's like you're splashing it with a very harmful chemical. But we do it because it kills bacteria as well. And so it has to be kept separate from the rest of the components of the cell. And that's what these somes, or these lipid vesicles, are great for, is that they contain peroxide. Yes. And what they break down, organic compounds for now, because it is a lot more complicated when we talk about peroxisomes, and we're going to remove that complication for your exam. In lab number 1, what do these do? Absorb. And so for lab number 2, there is these structures, these finger-like projections that cells will have on the surface of cells, not on all of not the entire cell, not on this part. You'll notice that there are no finger-like projections here, but you will see it on some parts. And so their role is to absorb things. So let's say that this is a cell that belongs in the digestive tract. We all have this concept. You consume food, it's going to go through your small intestines, travel along, and then the cells in contact with your food will absorb those yummy nutrients you absorbed, and your body's going to use it. Your lysosomes will break it apart into smaller pieces, and then your body will use it in order to carry out its metabolism. So microvilli are essentially just structures on the plasma membrane. So that's why we see the word membrane extensions. I would even just clarify this: plasma membrane extensions. Plasma membrane extensions that serve for the purpose of absorption. They increase the surface area for absorption. This can be a hard thing to understand, to increase surface area, but for us, and even understand it simply, it creates more absorptive space, more cells, more area for maximum or optimal absorption. And that's really what they're for. But for us, for our first exam, they just are for absorption, and that's good enough. But you will see, if you ever take a biology class, you will see this as a topic, surface area in cells, but that's not for our class. So I think we are almost done. Now, our cytoskeleton component. Recall that some proteins not only serve as molecular machines, but they also form filaments, or they form structures that will form or give integrity and structure to the cytoskeleton. And so if we look at our accompanying image here, we can see some of these components here. We can see cytoskeleton. Here, they drew it here at the edge for you. They sort of peeled off the plasma membrane so you can see it. And so here would be the cytosol, and in here you're seeing these protein filaments, sort of like a skeleton of the cell, providing some structure for the cell. And you can see, strengthens and supports cell in the same way that our bones strengthen and support the body. And then also remember that I said that they function like little transportation pathways, that if something's being delivered from the plasma membrane and it has to be delivered to somewhere along the body, it just has to be delivered somewhere, it uses them like little staircases, and then it'll actually travel and they'll actually deliver it wherever they need to go, and they use them like little roads or little pathways. And so that's why you can see that the complete definition includes some form of moving cellular structures within the cell. In that same light, when you look at this now, centrosome. There is only one of these. You're seeing right here at the very center here, there is our centrosome. Central sort of implying centralization, like the center of something. Some implying a vesicle. And so if you look at it, it's kind of in the center of the cell in a way. And it also has sort of a boundary around it, made more than likely out of lipids. So a lipid vesicle. And so what this does is that it is an organizing center because it can actually control things. It'll be attached to the organelles. It'll be attached to all of it, and it can move it wherever it wants throughout the cell, and it becomes more relevant for us when we talk about cell division. So what we're going to do is we're going to put a pin in it for now, and for now all you need to know is that it is capable of organizing all the inside of the cell in the same way that right now I'm seeing my desk here. I see my little book, my notebook here. I can put it here. This little sanitizing stuff, I can move it here. My paper towel thing, I can move it here. It's as simple as that. It moves all these organelles that we've learned about. It can move it wherever it wants. And where it will be relevant for us is that, let's say that we're making 2 cells. And let's say that I need to make this structure here, a Golgi body. If I'm making a brand new cell, and let's pretend I'm a cell, and I have my Golgi body. This is my Golgi body. And I know that I have to create another clone. There's going to be another Luis here soon because we're going to divide into a brand new cell or a brand new Luis. And so what does that Luis need? He needs a heart. He needs a liver. He needs the exact same things that I have now. And so what happens is that once the cell creates maybe another, or more than likely another Golgi body, it'll be the job of the centrosome to eventually grab it and move it to the other side, and then eventually, just use your imagination, Luis 1 divides to 2, and this organelle will now go to this other one because of our centrosomes. So simply, they just organize things around the cell. And that's all we really need to know about centrosomes. We'll discuss a slight detail about them further down the line.
Professor,
Ask away.
really quick so I can mentally prepare myself.
No worries.
It's what starts the mitosis or meiosis.
Yes, and that's exactly what cell division is, and we might actually get to it today. No, more than likely it's the beginning of Monday. But the word cell division, even if you want to get a head start on this for Monday, mitosis is the technical name for the process of cell division. The process of cell division. Sorry, my writing is terrible there. But the mitosis, the process of cell division. Looks terrible. Let's keep going. All right. I'm missing one organelle, and I'm not oh, here it is. And then the mitochondria. The mitochondria is what always jokingly usually is called the powerhouse of the cell. They're like the little batteries of the cell, and they produce ATP. This is a molecule that is considered an energy molecule. ATP. And that is their job. Energy molecule. And it is particularly unique because it also has 2 membranes. Everything else really only has 1 membrane, like 1 boundary, but it's complicated at the moment to probably think about it, but think about it like layers. Like right now you're seeing my skin, right? And so I have this shirt, this is 1 layer, and then I have this one underneath. So that's like 2 layers. It's kind of like, think about it like that. And there's even a space, like there's a space between shirt 1 and shirt 2, and there's activity going on in this space and activity going under the other shirt as well. So that's kind of a complication we're not really going to go into, but otherwise that's a unique thing about it is that it has a double membrane. All right, and that summarizes our cell, and nice. We're going to finish this whole packet today. And so, a talk about the cell wouldn't be complete unless we talk about the outer layer or the plasma membrane, the cell membrane, plasma membrane. And an important functional definition of it is that it is selectively permeable. Selectively permeable. We'll discuss that detail on Monday when we get back, or maybe at the very, very end. But otherwise, it means that it doesn't allow absolutely everything. And if you want to think about it right now, even simply, a human being fits through that door, right? This door selects for any organism that is approximately this big, right? If you're this big, it selects for you. You're selected for it. It's made for you. But if you're an elephant, good luck getting in through here, right? It is selective for humans, but not for elephants. The plasma membrane is the same thing, is that it doesn't allow particular things to go through, and it allows some things to go through. So it decides what it allows to go through. And the reason it has to be, or it's a very important property of it, is because it's not just like a dead wall like this of the cell. It's not just a container of the cell. It actually, if you use your imagination, like a ball pit, like a Chuck E. Cheese, right? If you were to like press down, you would go all the way to the ground, and you can hide in there, right, inside of the balls. That's kind of exactly what's going on with the plasma membrane, is that with enough force, if you imagine this was a plasma membrane, I would be able to put my hand through it and go to the other lab or the other room or whatever's on the other side of this, and then it would just seal around my arm, just completely enclosed. And just because I put my hand through it doesn't mean it's going to explode and all those organelles and cytosol is going to flow, but instead rather it just sort of encloses around whatever punctures it. You can grab a needle and you can actually place it inside, and you'll just see it open and then just collapse around the needle. And so it's rigid and tough, but it's also very bendable, flexible, and it has that dynamic ability to sort of retain the shape of the entire cell along with the cytoskeleton. And so it is going to control a few things if it is selective. It doesn't want everything going in through it. It definitely wants ions. What's an example of 2 ions we've learned so far? Potassium is one, yes, but sodium and chloride is one. We use the salt that we learned about earlier. And then our nutrients obviously have to go in there, our carbohydrates, our amino acids, things like that. It also has to allow for things to go in the opposite direction, right? Because we have to think about 2 directions here. And let's organize our photo. If we look at the top here, you can see extracellular outside. And here is the cytoplasm, or the intracellular fluid, but cytoplasm is easier. And so this outside space is separated from this inside space. And what do we think? I know it's already written there for you, but what do these look like to us? What organic compound? Lipids. And where's our tails, right? Do we see our tails? Inside here. So you can see how the tails organize themselves. So lipids not only make these vesicles inside the cytosol that are floating around, but they also form the plasma membrane. The entire plasma membrane, the entire boundary, is made out of lipids. And that's why fats and oils are so important in the body, or why they're an important part of a healthy diet, because you can't make any more cells without lipids. And so you don't have to flip pages, but I just wanted to show you why. We're going to come back to this very quickly. Remember that I said, what does the smoothie R? Oh, dang, it's already there. So what does the smoothie R make? Lipids. And if we're part of the ER or the rough ER, what's our next destination? The Golgi. And let's look at our definition of the Golgi. And the Golgi will store, alter, and package. And what does it store and alter and package? Lipids and proteins. And let's look at our illustration. Where are they going? They're going to the microvilli direction, or they're going to the plasma membrane, because even this, I know that it doesn't look like it, but these microvilli, this is a plasma membrane right here on the surface. All of this surface is a plasma membrane. The microvilli are nothing but extended plasma membrane, but so is this down here. This is also the plasma membrane. But these microvilli, the surface, are plasma membrane. And so doesn't it make sense that our Golgi apparatus stores, alters, packages proteins and lipids? And where do we see these lipids go? They go to the plasma membrane. The lipids belong on the plasma membrane. And proteins, more than likely, sometimes are also embedded in there. And you can even see in our little cartoon image here that these structures here are also proteins. You can even see they form some shapes that look familiar, right? The alpha helix is here, right? And so you can see that these are all proteins. So the Golgi apparatus delivered lipids and delivered proteins to the plasma membrane. And so you can see now full story, right? We're seeing why the Golgi apparatus delivers to the plasma membrane, and those free ribosomes are inside of the cytosol. They're creating things that stay inside the cell, but if you're a ribosome on the endoplasmic reticulum, you're traveling to the plasma membrane eventually. And so some quick little memorization, or like some properties, is that this outside layer is considered the aqueous layer, meaning the water layer. And so that's why we see, what is the name to love water? Philic. And to fear water? Hydrophobic. There is another property. You might see these little yellow ones. This is what cholesterol is for. Cholesterol is an important part of diets at some level because they actually contribute to the stability and they make it more rigid. You can actually make like a game out of this. Like if some cells have less cholesterol, they're a little more flexible and bendable. If you add more cholesterol, they turn more rigid and tougher. And so cholesterol is a component of the plasma membrane. And that's why cholesterol is an important part of the diet, but obviously particularly in America, a lot of things are so heavily rich in cholesterol that it causes actual problems, sometimes more than it actually helps. So with that, I'll let you all go 15 minutes earlier. So let's keep going just slightly more because I know your brains are probably wearing thin right now. And so anything I want to touch on today that I really want you to leave with the weekend, we got our cholesterol. We know that they're made out of phospholipids. And what were the other types? There's phospholipids and there was one more type of lipid. What was the other one? Oh, that's proteins. We'll get into that one next. But glycolipids, phospholipids. So phospholipids is the main component, but now we still haven't addressed where these come into play. Recall that they do the same thing. They have the tails and they embed themselves. So these are called glycolipids along with your phospholipids. And so if you look at this, what do we see? We see our this is wrong. This is a publishing error. This arrow should actually be pointing to this. This is not a glycolipid. This is a phospholipid. So this is a publishing error. Try to draw a little X here to remind you of that.
Or just scribble around the dot.
Yes, whatever. Oh yeah, you can scribble it out and rewrite it and then write another arrow. That's true too. And so here's a perfect example of a glycolipid because here you're seeing the water-loving portion. This is a full carbohydrate chain. Remember that you're seeing the little hexagon shape, the little glucose shape as an example, just a rough example. It might not be glucose or it might be, but otherwise you can see that we have a sugar on top. And what do? Their job is to serve as recognition sites. They are little flags that saying, hey, I'm an intestine cell. Hey, I'm a nervous cell. They all have a unique sugar profile on the surface. If you look at it as a whole, if you just examine the entire sugar profile, different cells have different profiles. An even better example is with white blood cells. White blood cells are swimming in your bloodstream and they're always looking for dangerous things flowing in your bloodstream. And let's say a little bacteria is inside there. Inside of those bacteria also have little, exactly like this, they have little profiles of sugar at the top. And so your white blood cells are always examining your own cells. They're like, you're good, you're good, you're good. And then they see a profile they don't recognize, like a sugar profile. Again, the sugar profile is called a glycocalyx as a whole. And so the whole profile will be examined by the white blood cell. It'll say, oh, you're good, you're good. And then finally a little bacterial one, it touches it and it says, whoa, whoa, whoa. I don't recognize this sugar or this sugar surface. And then it says, destroy it right away. And that's what the glycocalyx is for, is that it's sort of a profile for the surface of a cell. And so your body is also investigating. These can change according to the health of a cell. If a cell is telling it's time for the cell to die, it might actually it's a complication that there's like through multiple angles to that I don't want to address. But that's what a glycocalyx is that it is the sugar molecules on the surface. And do me a favor and get rid of all of this. You don't need this. Yes, get rid of all of this. You don't need this. All you need to know is that glycocalyx is a sugar profile on the surface of cells. Oh, dang it.
Professor,
Ask away.
really quick, I saw this really crazy
Mm-hmm.
And does bacteria, so how it works is like once it's in the body after a certain amount of time, the bacteria changes its recognition. I don't know if it's also glycocalyxes, but you know how like they have the little things that stick out from the bacteria, so they change their DNA while they're in your body, so your white blood cells can't recognize them anymore.
Okay, I won't use bacteria because my microbiology is too. I don't remember anything anymore for microbiology or some of those properties, but I will say I was a virologist and I do understand viruses. So, to that same effect, and I guess I'll end the class on that fun fact since COVID was such a, something affected us all, is that viruses actually hijack those. That's why some viruses are useless in other places, and in some places, COVID, for example, like it looks and loves the cells inside of your airway. And so it actually uses it and it says, oh, this is, and you learn this too. What is the, I don't want to say any more time, but otherwise the cells, the pseudostratified columnar cells that you learn about in lab, I know that, I'll mention it again next week for lab number 1 because I know we went over it. Lab number 2, I'll talk about it today with you today. But otherwise, viruses will say, oh, this is my favorite cell to target, and it uses them as a recognition site, and then they're able to hijack the entry system because their surfaces change. And so viruses will come, hijack this, and then they're gonna hijack your ribosomes, and then they're gonna produce their own components, and they're gonna use your own ribosomes to make their own proteins, and they're gonna form inside, and then eventually, finally the cell will eventually realize it and then decide, oh, I need to destroy the cell, I'm infected. And then, but it's sort of a double-edged sword because once the cell destroys itself, 1 million viruses, it only takes one virus to hijack a whole cell, and then it can produce millions in a matter of an hour. And then all of a sudden, all of those will go and infect the rest of the cells that are adjacent to the one cell that we're seeing in this particular illustration. And so viruses are, think about this, is that if you were to, but when you place in viruses inside, one virus goes in, out of 1 million, even though you have all these white blood cells and they're killing all these viruses, once they realize it and they're attacking it, there'll always be a virus that was formed incorrectly. Its DNA was formed incorrectly, but that little incorrection caused it to create a recognition site or a surface that's different from the rest of it. In other words, it actually became deformed, and then your own system can't recognize it as dangerous, and so then it's because it's deformed, then it can just create and infect your cells even though your body's trying to fight the virus off. And then, when we get to white blood cells, I'll add that little talk about it with some better pictures because, trying to make you all visualize this is, I think, tough. But I'll introduce some cool visuals next time that we can actually use to talk about that. That would be kind of fun to add. And so we will pick up here next class. And I believe the lectures for tissue is already up. So if you want to get a head start on Monday and you just want to keep studying tomorrow, Saturday, or Sunday, those lectures are up. I'll provide those links. Just give me till tomorrow because I am busy with training stuff until 11:30 tonight. But I will try to do it during lab. There was no time in lab. We'll try to, tomorrow, guaranteed, I will have them all up around 11 a.m. Everything will be organized on Canvas so you can find the videos faster for the remaining bit of the material. All right?